channel. And the recording is going to be uploaded onto the clinician resources site. You can see the QR code for that here. And I'll send out more information on accessing the recording after the session. One other reminder, it looks like everybody's muted, but if you all could stay muted unless you have a question, we would greatly appreciate that. So without further ado, it is my great pleasure to introduce today's speaker, Dr. Louisa Messenger. So Dr. Messenger is currently an assistant professor in the Department of Environmental and Occupational Health at the University of Nevada, Las Vegas School of Public Health. Before joining UNLV, she was an assistant professor at the London School of Hygiene and Tropical Medicine and had more than 14 years of experience specializing mostly in molecular parasitology and entomology of vector-borne diseases across the world. Dr. Messenger has made substantial contributions in two main research areas. These include the development and evaluation of innovative vector control interventions, as well as the application of basic science techniques to improve disease surveillance and management strategies. Specifically, talking about Chagas disease, Dr. Messenger did her PhD in Trypanosoma cruzi molecular epidemiology from the London School in 2014. She's very passionate about improving our understanding of T. cruzi genetic diversity and its implications for the clinical, ecological, and biological aspects of Chagas disease. So she's going to tell you guys all about this today. So without me going on any further, Dr. Messenger, I give you the floor.
Thanks so much, Eva. So hi, everybody. And I just want to start off by saying a really big thank you to the US Chagas Disease Providers Network, so to Eva, Karen, and Jeff for inviting me to talk to you today. So I'm going to walk us through some of the perils, pitfalls, and possibilities in historical and contemporary genotyping of Trypanosoma cruzi. And by the end of this talk, hopefully you will all be as enthusiastic about T. cruzi genetic diversity as I am. So starting with a quick overview, I'm going to present a brief history of Trypanosoma cruzi taxonomy, as it's really important for us to understand how we got to our current nomenclature and classifications within this parasitic species. I'm then going to tell you all about Trypanosoma cruzi genetic diversity, its evolutionary history, and methods of genetic exchange. Then, once you have a better understanding of some of the genetic complexities underlying T. cruzi, we're going to talk all about T. cruzi genotyping and some of those perils and pitfalls. And then finally, I'm going to end with our contemporary possibilities and potential shifts in the paradigm of T. cruzi classification. So starting with a brief history of T. cruzi taxonomy. Now, the earliest reports of genetic diversity in T. cruzi, they date back to the late 1970s, where the most advanced laboratories with molecular capacity were using multi-locus enzyme electrophoresis, MLEE, for classifying parasites. Essentially, this technique is really similar to agarose gel electrophoresis, which we routinely use to separate out our PCR products, and I'm sure many of you will be quite familiar with that technique. Instead, with MLEE, here each organism is differentiated by the relative mobility of different intracellular enzymes that are separated under an electrical current. And Michael Miles, my former PhD supervisor, was working in Brazil using this technique to analyze different T. cruzi isolates derived from humans, from mammalian reservoirs, and from terratinine bug vectors. And initially, these were classified on the basis of MLEE into three groups, or zymidines, called T. cruzi 1, T. cruzi 2, and T. cruzi 3. Then, in 1981, this is when things began to get really interesting. Michael and his research team analyzed a series of Chagas disease clinical specimens and published a landmark paper in The Lancet, which reported distinct geographical differences in the clinical presentation of Chagas disease, which in turn were linked to the different zymidines of T. cruzi. And that's this landmark Lancet paper from 1981. You can see here on the left, this shows you what those multi-locus enzyme electrophoresis gels look like, where intracellular enzymes are separated under electrical current to produce these distinct banding patterns. And what this paper reported, which was so significant at the time, was that among 316 T. cruzi isolates from Venezuela, from the Brazilian Amazon, and from Central and Eastern Brazil, T. cruzi appeared to be genetically different on the basis of these multi-locus enzyme electrophoresis profiles, such that up in Venezuela, where clinicians rarely saw cardiac disease or gastrointestinal disease, Trypanosoma cruzi zymidine 1 was implicated as the major circulating parasite strain. While further down south in Central and Eastern Brazil, a different zymidine, T. cruzi 2, was the causal agent. And here, T. cruzi infection more commonly presented as digestive megasyndromes. And so it was this clear geographical split by parasite genetics and clinical presentation, particularly that predominance of gastrointestinal symptoms in Southern South America, that then became canon for several decades after. And this concept of distinct parasite strains inducing different clinical pathologies was a widely accepted dogma that has persisted and somewhat plagued genotyping efforts with T. cruzi to this very day. And I'll get into that in a little bit later in the talk. But I just want to recognize that this paper was a real game changer at the time. It did, of course, suffer from reporting bias. It only used parasite strains derived from symptomatic patients, as well as was heavily limited by the lack of resolution that you can achieve with this particular, now antiquated genotyping technique. This brings us a little bit more forward in time, 1988. By this stage, multiple research groups were working with as many different T. cruzi strains as possible, mainly using multi-locus enzyme electrophoresis. And this enabled researchers to further subdivide parasite strains into more than 43 different planets or subdivisions of these major 3 zymidines. And these are some of the earliest papers, which, while not using a very resolutive genotyping technique, they were generating some of the first observations of just how genetically diverse a parasite T. cruzi really is. And that's this paper here from 1988, which really began to probe at the levels of genetic diversity in different T. cruzi strains. Again, this is one of these multi-locus enzyme electrophoresis gels showing different banding patterns. You essentially perform this analysis by scoring your banding patterns across multiple enzymes. You can see that in this table here. So at the top here, these are multiple different T. cruzi enzymes. So GPI, for example, is glucose 6-phosphate isomerase. G6PD is glucose 6-phosphate dehydrogenase. I won't name all of these enzymes, you get the idea. But then essentially what you do is you score your band numbers based on how fast they migrate in the gel. So the closer they are to the bottom of the gel, the faster they migrate. Each of your individual strains essentially has this unique barcode, and you can then use that information to construct Wagner networks down here. Essentially, we're trying to reconstruct the relatedness between parasite strains. You can think of these networks as early precursors to haplotype networks or phylogenetic trees. But really the take-home message from these early studies was just how genetically diverse and distinct so many different parasite strains were from one another. Moving forward from 1988 to 1999, multiple studies using multi-locus enzyme electrophoresis were published, trying to classify, subclassify, reclassify, and characterize different parasite isolates. And because T. cruzi taxonomy at the time was highly unstandardized, in 1999, a consensus meeting was convened in Rio de Janeiro by Chagas experts in an attempt to basically define working parasite nomenclature. And this expert panel agreed at a minimum that parasite isolates could be classified into 2 major genetic lineages, T. cruzi 1 and T. cruzi 2, on the basis of several multi-locus enzyme electrophoresis studies, and then also other reports using some of the earliest DNA markers for this parasite. Then a couple years later, in 2001, that second lineage, T. cruzi 2, was further subdivided into TC2A, 2B, 2C, all the way up to 2E, on the basis of more advanced DNA-based genotyping methods, principally multi-locus sequence typing, or MLST. And that's this paper here from 2001. Multi-locus sequence typing is really similar in premise to multi-locus enzyme electrophoresis, but instead of examining the mobility of different parasite enzymes, instead here you essentially use different DNA-based markers instead. You use multiple parasite genes to produce a unique barcode per sample. And here you can see some of the most common genetic targets that we routinely use to characterize T. cruzi isolates and their different banding patterns. So here in this decision tree, you take your T. cruzi isolate. We first PCR up the mini exon. This is the spliced intergenic region. It's a non-coding intergenic region, which can be quite variable for a nuclear gene. If we see a PCR band of 350 base pairs, that may be indicative of T. cruzi 1. Or if we see a band of 300 base pairs or no amplification at all, then we move on to our second PCR, the 24S alpha ribosomal RNA. Again, based on our banding pattern and our sizes and our numbers of bands, this may indicate TC2D, TC2C, or 2A, or alternatively, we may go on to our third assay and PCR up the 18S ribosomal RNA to distinguish between TC2B and TC2E. Now, this paper is an early version of multi-locus sequence typing, but we still frequently use some of these PCR assays and these types of decision trees to genotype T. cruzi nowadays. Really depends on the type of available sample that you have, as well as your laboratory resources. Fast forwarding a few years later to 2005, this was really the stage at which we began to truly understand just how genetically complicated T. cruzi really is. And this was when the first whole genome sequence of the strain C.L. Brenner was published in a landmark Science article. Now, the first genome of T. cruzi C.L. Brenner was published in 2005, but this initiative actually began back in 1998. One of the reasons that it took so long is because C.L. Brenner is actually a hybrid strain of T. cruzi. We currently recognize it as a member of the TC6 lineage, which I'll tell you about in just a moment, but it's a hybrid of a recombination event between TC2 and TC3. But without knowing it was a hybrid strain, it was selected to be the first whole genome sequence using 454 shotgun sequencing, mainly because it's one of our most well-studied and well-characterized strains in the laboratory. It was originally isolated from a triatamine bug in Rio Grande do Sul in Brazil. Now, in terms of the size of our genome of T. cruzi, it's between 55 and 60 megabases. It's almost twice the size of the genomes of closely related medically important trypanosomes, like Trypanosoma cruzi and Leishmania zonavani. And because of its hybrid nature, that made the whole genome shotgun assembly of C.L. Brenner really fragmented, mainly because of the large number of allelic polymorphisms present because of its hybrid nature, and because a large proportion of the genome is made up of repeat regions. In fact, close to 50% of the entire genome is composed of repeat regions. And you can see some of these large gene families composed of repeat regions here in this table. The largest of these families are the transialidases with at least 1,400 members, then our mucine-associated surface proteins or MASPs, and then other types of mucines. And these families, they play really important functions in host-parasite interactions on the cell surface of T. cruzi, possibly allowing the parasite to evade the host immune response and to mediate tissue sequestration. However, these families of repeat regions with their thousands of members, they make it virtually impossible to correctly assemble these parts of the genome. So our whole genome sequences for T. cruzi that are available and assembled, they really represent the non-repetitive regions, of which we've identified about 23,000 genes, you can see here. Half of these still, though, have no functional annotations. And it's the absence of a completely assembled reference genome for T. cruzi, which has also been a real limiting factor for subsequent comparative genomic analyses. When you compare where we are with T. cruzi to other parasitic trypanostids, we're somewhat falling behind. This brings us now to 2009. By this point, multiple DNA markers supported the classification of parasites into at least 6 major genetic lineages. These are the same that we had previously, TC1 and TC2A to E, but this time we wanted to remove that presumptive designation that there was 2 major groups, and then it was a second one that was subdivided. So our lineages were renamed to be TC1, TC2, all the way to TC6. We also call these DTUs, or discrete typing units, and this is the current T. cruzi nomenclature that we recognize and we use today. And this is what our current understanding of T. cruzi genetic diversity looks like. Now, generally, each T. cruzi lineage has a broad but often overlapping geographical and ecological distribution. TC1, TC2, TC5, and TC6 These are frequently isolated from domestic transmission cycles, and they're responsible for the majority of human infections. The distribution of TC1 extends from the Amazon basin northwards, where it's the principal cause of Chagas disease in Venezuela and Colombia. TC1 is also ubiquitous in arboreal sylvatic transmission cycles throughout Latin America, primarily circulating in ecotopes between Didelphis opossum species and Rodnius triatomine bug vectors. TC1 also has secondary terrestrial transmission cycles among rodents and sylvatic triatoma species in the inter-Andean valleys of Bolivia, Peru, and Chile. Now with TC1, we've got multiple molecular markers that consistently identify high levels of genetic diversity within sylvatic parasite populations, and then divergent but genetically homogeneous strains associated with human infections. By comparison, TC2, TC5, and TC6, they're less genetically diverse overall. They appear largely confined to domestic transmission cycles in southern parts of South America. The sylvatic reservoirs of these three lineages are not fully defined, although TC2 has been increasingly isolated from primates in Brazil, and TC5 and TC6 have also been isolated from dogs, peridomestic dogs, as potential reservoir hosts in the Gran Chaco. TC3 has a dispersed terrestrial distribution that ranges from Amazonia down to Argentina. Here it's principally transmitted by Panstrongulus triatamines to Dasypus armadillos and other burrowing fossorial mammals. TC4 is comparatively poorly understood, mainly because several different genotyping methods fail to distinguish this lineage from others, mainly from TC3. However, TC4 is known to circulate sympatrically with TC1 in wild primates and in monodelphis opossums and Dasypus armadillos in the Amazon. And raccoons and dogs up here in North America, we also frequently see TC1 and TC4 co-infections. In terms of its importance for disease, TC1 is increasing in its importance as a secondary agent of Chagas disease in Venezuela. It's also been isolated from recent oral outbreaks in the Brazilian Amazon and Colombia. And then finally, we've got TC-BAT. TC-BAT is the newest genetic lineage, the seventh lineage, first described in 2009, and it's so named because it's been isolated from bat species almost exclusively across Brazil, Panama, Colombia, and Ecuador. To date, there's just been one isolated human case report of this lineage in a child from Colombia. Now, in terms of the evolutionary history of T. cruzi, in general, molecular analyses indicate that T. cruzi has a predominantly clonal population structure that's been interspersed with infrequent genetic exchange events, which have shaped the evolution of this parasite. What this really means from a genotyping point of view is that depending on marker choice, most parasite strains can be assigned to one of these 7 lineages, but there is quite substantial genetic diversity between parasite isolates that belong to the same lineage. On this slide here is one of the most widely accepted models to describe T. cruzi's evolution. TC1 and TC2, these are the oldest genetic lineages. They diverged from each other about 4 million years ago. A hybridization event that occurred about 2 to 3 million years ago between these two lineages here, it formed TC3 and TC4. Then, much more recently, about 60,000 years ago, concomitant with the dispersal of humans across the Americas, at least 2 genetic exchange events between TC2 and TC3 produced TC5 and TC6. And isolates of TC5 and TC6, they have all the characteristics of new hybrids. They're highly heterozygous, and they possess intact alleles from their parental progenitors, TC2 and TC3. Now, in terms of the mechanisms of genetic exchange, with improved sampling strategies and the development of higher resolution nuclear and mitochondrial genotyping techniques, there's a growing number of field studies that indicate that natural recombination in T. cruzi may be more frequent, it may be non-obligatory and idiosyncratic, potentially involving independent exchange of nuclear and mitochondrial material, as well as potentially canonical meiotic mechanisms. And you can see the different types of mechanisms of genetic exchange on this slide. So at the interlineage levels, for example, TC5 and TC6, they're unequivocal hybrids. They resemble diploid, heterozygous Mendelian F1 progeny, showing intact alleles from their parents, TC2 and TC3. but we don't necessarily see evidence of all lineages undergoing meiotic recombination. Rather, in a number of field populations, what we see instead is widespread mitochondrial introgression, where essentially our kinetoplastid DNA is being exchanged in the absence of any nuclear recombination. Now, we see this between strains of the same lineage, like TC1, as well as between major DTUs like TC1 and TC4, and TC3 and TC4. Now, one possibility for this occurrence, the independent exchange of mitochondria in the absence of nuclear material, it may be due to the crucial role that the mitochondria plays in growth, development, and metabolism, such that asymmetrical mitochondrial integration may satisfy the elevated need to escape Muller's ratchet, that being the irreversible accumulation of deleterious mutations that results from clonal reproduction, which is the predominant reproductive strategy of T. cruzi. But suffice to say that mitochondria moving, sometimes in the absence of nuclear material, this just serves to complicate parasite genotyping endeavors. The last genetic exchange mechanism, which we've only ever demonstrated in vitro, is parasexual fusion, followed by progressive stochastic genome erosion. And I'll show you this in more detail now. So in 2003, this landmark Nature paper was published, where the group at the London School of Hygiene and Tropical Medicine were able to produce hybrids between TC1 strains. You can see here in these gels, this shows you parent 1, parent 2, and then their hybrid progeny in the middle that possess intact alleles distinctly from both parents. You can see in both of these. But these hybrids, they weren't formed by canonical meiosis. Rather, parts of the genome appeared to be triploid or subtetraploid. You can see that here in these microsatellite traces. This is parent number 1, parent number 2 at the same genetic locus, and here's our triploid or subtetraploid progeny here, possessing all alleles from both parents. So after this Nature paper came out, the same study team really wanted to further understand what happens to these subtetraploid hybrids at the genetic level over time. And I really just want to highlight this paper here as one of the more ambitious studies that we've really ever attempted when it comes to T. cruzi genotyping. Because what we did to understand these hybrids was we maintained these T. cruzi hybrids in vitro culture for 800 generations. That's a little over 5 years. So we as a team, 4 of us as parasitologists, we essentially subcultured our parasites consistently for 5 years' time. We did this without losing any of our cultures to contamination, which is nothing short of a miraculous feat. You can see the experimental setup here. Essentially, we subcultured our parasites every 2 weeks for 5 years, and then we took longitudinal samples, and then we did whole genome sequencing to understand those patterns of genome erosion in comparison to our reference genome. And you can see these patterns of genome erosion across 3 different chromosomes here at the bottom. This shows you copy number variation across these different chromosomes, showing you the parents after the initial hybridization event, then after 5 years of culture. Similarly, our hybrids initially and then after culture. And you can see stochastic random genome erosion across these different strains over 5 years at different points in the genome. These hybrid strains, even after 5 years of continuous culturing, they still haven't returned to diploidy as they would be expected to do if they had undergone a canonical Mendelian genetic exchange event. Okay, so at this stage, just to recap, the real take-home messages that I hope have impressed upon you are that T. cruzi is a highly genetically diverse parasite. It's had a complex evolutionary history, and there are multiple different mechanisms of recombination that happen in the lab and the field, all of which essentially serve to complicate our ability to reconstruct phylogenetic relationships between parasite strains and to make causal associations between parasite genotype and various clinical and ecological features of Chagas disease. Now we're going to step it up a gear and we're going to get even more complicated. I'm going to talk to you about our parasite genotyping perils and our pitfalls. Because up until now, I've simply been referring to T. cruzi parasites as isolates, strains, or clones, but in reality, each infection is actually composed of a multi-clonal parasite population. In the natural environment, susceptible hosts experience repeated contacts from multiple infectious triatomine bugs, which in turn may have fed on a range of infected humans and/or mammals, depending on the local disease ecology. Furthermore, in both adult humans and mammalian reservoir hosts, chronic T. cruzi infection is rarely cleared by chemotherapy, leading to an accumulation of genetically distinct parasites within an individual. These transmission dynamics, they render parasite multiclonality, i.e. an intra-host or bug infection comprised of these diverse parasite clones, as really being inevitable rather than exceptional with this parasite species. Now, this slide here is a really crude representation of T. cruzi multiclonality, but basically an individual human or animal reservoir or triatomine bug will not be infected by just that one single genetically homogeneous parasite, but rather these multiple clones that make up this multiple clonal parasite population. And the genetic composition of this population in an individual host is going to be determined by a number of different factors and will likely be in constant flux. The triatomine bug here is going to ingest a subset of the multiclonal T. cruzi population from an infected person or mammalian reservoir host, the part of the parasite population that's circulating in the person or the animal's peripheral blood at that given time of blood feeding. As this multiclonal parasite population develops within the triatomine bug to the infective stage for transmission, there will likely be bottlenecks of selection for particular clones, as well as de novo mutations imposed by that triatomine's immune response to the parasite and by competition between parasite strains for finite nutritional resources in the bug. Now, then, depending on the bug's feeding behavior, whether it was able to obtain a complete blood meal the first time it fed on a host, or if it refeeds during this parasite development period, the genetic diversity of its multiclonal parasite population may increase or change as it takes additional blood meals from hosts which have their own genetically distinct multiclonal populations. Now, coming on to our human, estimating parasite diversity is clearly going to be complicated by sequestration of parasite clones in tissues as the mastogote stage of the life cycle. And in humans and animal hosts, the extent to which distinct parasite clones have proclivity for circulating in the peripheral bloodstream or sequestering in the tissues, we really don't know. It's poorly understood. Now, in our animal or our human, there will be a proportion of genetically distinct parasites circulating as our tissue-derived trypanosomes in the blood. These may be quite different from our amastogotes in the tissue. And again, we would expect our levels of intra-host genetic diversity to be constantly changing from selection by the immune response, our rate of tissue sequestration as the amastogote stage, a steady mutation rate, intraparasite competition, such as genotype displacement, reciprocal inhibition, potentiation, inbreeding, or even recombination via those genetic exchange mechanisms that I've shown you. And then finally, of course, our force of infection from infectious triatomines as well. So what does this really mean when we come to try to genotype T. cruzi? Well, we do have a number of different types of molecular assays at our current disposal. Many of them have inherent limitations, and most of the time we're really just scratching the surface of exploring this parasite's genetic diversity in particular host and transmission cycles. In general, choice of genotyping technique depends on the source of the biological specimen, and then obviously the research question, the funding, and the laboratory capacity. If we start here with our clinical specimens, these are from humans, animal reservoirs, or triatomine feces, and it may include blood, tissue, cerebral spinal fluid, or serum. The first issue that we encounter is that the volume of these types of specimens is usually really small. And for chronically infected adult patients, peripheral parasitemia is very, very low, often or not. It may be less than 1 parasite per mil of blood. So with a low starting material and an abundance of other host DNA in the sample, it can be really difficult to do any type of genotyping due to assay sensitivity and inhibition. Sometimes the best that we can do is simply the presence or absence of T. cruzi based on quantitative PCR. For those studies that do try to do some genotyping for clinical specimens, in general, our assays are only sensitive enough to identify major T. cruzi lineages. And hopefully you can appreciate from the previous multiclonality slide, this is obviously a gross underrepresentation of true parasite genetic diversity within a host. These genotyping techniques that we use for clinical specimens, they're almost always based on multi-copy gene targets to improve our assay sensitivity. They're usually PCR-based techniques like restriction fragment length polymorphism or amplicon size polymorphism. And they'll almost always detect the predominant genetic lineage or lineages amongst our multiclonal population. Now, because of all of these issues with clinical specimens, the workaround that a lot of us try to use is to try to isolate parasite strains by seeding some of our clinical or biological sample into culture. Now, this gives us the advantage of being able to grow up large quantities of biological material, and it also allows us to isolate some of these genetically distinct clones from our multiclonal population by doing serial dilution cloning or by doing FACS cell sorting. The issue we have with this, though, is that the recovery rate is really, really low. Even if you start with large quantities, say something like 30 cc's of blood, we often recover less than 20 to 30% of our parasite strains. And of course, we as researchers, we are then imposing a massive genetic bottleneck or selection for particular parasite clones that grow well in our specific culture media. And then, of course, we're also only growing parasite strains that are circulating in the peripheral blood to begin with. But because we can potentially maintain these strains in culture long term, this then does allow us to do higher resolution genotyping, potentially to the intra-DTU level, depending on our genetic marker choice. Now, conventionally for genotyping assays, we either use the nuclear or the mitochondrial genes, and oftentimes we'll use a combination of both. In terms of our nuclear genes, for example, for multi-locus sequence typing in particular, we often use single copy housekeeping genes, but these by definition have very limited sensitivity, especially if your starting material is low. And this is because these genes, they serve an essential function in T. cruzi, and so they're reasonably well conserved even between major lineages. Now we can use multiple copy genes. I showed you some of those, like the splice leader intergenic region earlier, and these are more sensitive because they are multi-copy in nature. But if you're dealing with a multiclonal parasite population, it's impossible to distinguish what is a multiclonal parasite population from diversity across multiple copies of a gene within a single clone. That is, unless you start with a single clone as your starting sample. Now, with our nuclear targets in general, because coding genes are fairly well conserved, we're really only able to identify major DTUs. It's harder to investigate parasite genetic diversity within DTUs. To do this, we can use multi-locus microsatellite typing schemes. There are a few available to do high resolution population genetics, but these are usually laboratory specific. They're not portable or transferable between different sequencing machines, and they're not really used routinely. In terms of our options for mitochondrial genes or our kinetoplastid genetic targets, the T. cruzi mitochondria is composed of about 20 to 100 copies of maxicircles. These contain the coding genes. They're about 20 to 40 kilobases in size. These are concatenated in a very dense network surrounded by several thousand much smaller mini circles. Now, we can use fragments of the maxicircle for genotyping. These regions mutate about 10 times faster than nuclear genes, and they do so because of the production of reactive oxygen species and much more limited DNA repair mechanisms. So these genes can facilitate higher resolution genotyping, potentially elucidating genetic diversity at the intra-DTU level. But as I already mentioned, mitochondrial integration is a fairly common mechanism of genetic exchange, and that complicates the use of these genes for phylogenetic reconstruction, particularly if they're used in isolation without any nuclear targets. Now, there are some research groups that use mini circles for genotyping. They use probe hybridization to the hypervariable region of the mini circle to produce highly variable strain-specific profiles. The problem we have, though, with this technique is that these probe assays are really difficult to reproduce between laboratories and even between technical replicates of the same parasitic strain. And also then the interpretation can be quite subjective as a result of this. Now, looking more specifically at the clinical context of T. cruzi genotyping, which is probably of the most importance for most of you listening to this seminar, here's the bottom line. Only about 30% of chronically infected Chagas patients will test positive by quantitative PCR, simply because our circulating parasitemia is just so low. It really does limit our ability to do any reliable genotyping. This table here, this shows you just how variable and how low some of these circulating peripheral parasitemias can be. This shows you patients from Brazil, from Argentina and Colombia, and here we're talking less than 1 parasite per mil. Now, these levels of parasitemia, they are detectable by qPCR. There are a few assays out there that have attempted to use melt curve analysis with qPCR assays to do genotyping of clinical specimens, but this is only really ever to the DTU level. Now, furthermore in studies, though, others have attempted, for example, to do longitudinal genotyping. This opens up another caveat. So here, this table here is from Colombia, from patients there where they had their blood drawn every 10 days for genotyping. And we see fluctuations in our major predominating parasite lineages, which is to be expected if we think back to the biology of this particular parasite. Here in this table, for example, we see changes in the proportion of T. cruzi 1 between our first blood draw, our second blood draw, and our third blood draw. Other observations, we see TC4 present in our first blood, is completely absent in our second and third time points. Similarly, we see co-infections between two distinct lineages in the first blood draw that's completely absent in the subsequent 2 time points as well. I really just want to emphasize that our peripheral parasitemia populations are so highly dynamic. And on top of that, we still have that unascertainable amount of parasite genetic diversity that's sequestered in the tissues as amastogotes. Now, in terms of all of our available genotyping techniques, this table here, this shows you a summary from a recent review article of all of the different available methodologies for T. cruzi genotyping, and then the total number of specimens that have been genotyped using these particular techniques in the published literature. I'm not going to go through all of these. I simply want to make the point that we do have a reasonable amount of available options. They're designed around different assay modalities. Again, they'll have different levels of resolution to greater or lesser degrees. Really depends on the underlying biology of the type of genetic marker, and then on the type of starting biological sample that you're trying to genotype with T. cruzi. Now, those are all of the numerous inherent limitations associated with the genotyping T. cruzi. But I want to bring us back to why are we so interested in doing this in the first place? And it really began with that Lancet paper in 1981. In terms of Chagas disease clinical presentation, we certainly see differences in the prevalences of cardiac disease versus gastrointestinal manifestations. And of course, only 30% of our chronically infected patients, they ever present with symptomatic clinical disease. So it's really tempting to implicate parasite genetic diversity as the causal drivers of these phenomena. But is that really the case? And is it ever going to be as simple as a particular parasite clone is responsible for clinical disease or cardiac versus mega syndromes? Unfortunately, unlike some other parasitic species, it's really unlikely to ever be that straightforward with T. cruzi. Nothing is straightforward with T. cruzi. Back in 2015, myself, Michael, and Karen, we went through all of the published literature extensively to try to evaluate the evidence basis for those causal associations between clinical aspects of Chagas disease and parasite diversity. And what we concluded from this literature review was that parasite strains that we detect in our patients, with or without morbidity, it really just reflects what the principal lineage that's circulating amongst domestic transmission cycles is in that particular geographical area. And that what we really need is improvements in both our study design, particularly the investigation of parasite genetic diversity in symptomatic as well as asymptomatic patients. And then we also need parallel improvements in our genotyping techniques. They're really required to advance our understanding of the role that T. cruzi can play in disease pathogenesis. Now, in terms of improving our study design, some of the best Chagas disease patient cohorts with the most extensive clinical workups, they're found in our drug trials. But unfortunately, because of the limitations that we experience with T. cruzi genotyping, the amount that's been done in clinical trials is really limited. This slide just shows you some of the most recent large-scale clinical drug trials for Chagas disease. And most of the time, no parasite genotyping is ever attempted. And frankly, I don't blame them. If any is, it may be assumed by geography at best. And again, that's not very informative. You can see here the Bendita trial, here's the Benefit trial. Both of these didn't do any actual genotyping. They just assumed the predominant lineage based on the geographical origin of their patient cohorts. But as I've already mentioned, there is considerable geographical overlap between lineage distribution. And really looking at the DTU level simply isn't enough to start to try to link parasite genetics with any clinical outcomes or drug efficacy. Similarly, the most recent drug trials of ravuconazole, E1224, they also just didn't even attempt any genotyping either. But are we really expecting to see differences in T. cruzi genotype according to drug response? Well, the literature is really mixed. Most of it's based on in vitro drug assays and using lab cultures. So obviously there are inherent barriers and drawbacks to that in itself. On the left here, this just shows you a recent meta-analysis of susceptibility of T. cruzi laboratory strains representing major DTUs to benzonidazole. And you can see here across our epimastogotes, across our tissue-derived trypanosomes, and across our amastogotes, we see quite a large amount of variability in the lethal dose 50 for benzonidazole. You can also see that heterogeneity in parasite response here manifested in these funnel plots. Essentially, they're just highly variable. There's no clear trends in our LD50 to benzonidazole by T. cruzi lineage. Rather, we just see strain-specific variability. Similarly, here on the right-hand side, this shows you the efficacy of 3 parasite strains, a TC1, a TC2, and a TC5, to nifurtimox, benzonidazole, and posaconazole. Again, showing you variability in drug response by genetic lineage and also inconsistent variability by lineage. For example, our TC5 strain, it sometimes seems to be quite responsive, sometimes much more less responsive, depending on the compound under evaluation. And then when we begin to look at other clinical aspects, again, we do see clear differences, for example, in T. cruzi diagnostic test sensitivity, which may be partially related to T. cruzi genotype, but again, we don't see any clear one-to-one associations. The papers on this slide, they highlight one issue that we certainly see here in the US, and that's this differential diagnostic test sensitivity. We see these differences in serological test sensitivities between patients from Northern South and Central America compared to patients from much further south in Latin America, which there we're interpreting as being related to the predominance of different T. cruzi DTUs. And there is some good evidence for this at the antigenic and the diagnostic assay level, but it's indirect evidence for the most part. On the left here, though, this is a great recent paper showing you variability in seroreactivity to different regions of the T. cruzi proteome. So sera taken from patients from different endemic regions. You can see Bolivia, the US, Argentina, Brazil, Colombia, and Mexico. We certainly see differences in clusters of private antigens per geographical populations, which may be indicative of a link to our parasite genetics and may explain some of our differences in test sensitivities. On the right here, we're seeing the same phenomenon, but at the serological test level. You can see the proportions of patients being diagnosed on multiple serological tests vary significantly between Argentina, between Honduras, and between Mexico. And again, this is potentially indirect evidence that the influence of parasite genetics can play on test sensitivity. But really, where does this leave us? And really, what are the future prospects for research in this particular area? Well, clearly based on our historical genotyping techniques, we're not getting anywhere in terms of truly characterizing parasite genetic diversity or establishing any causal links between parasite genotype and other aspects of clinical disease or parasite diversity. But there have been some quite remarkable advancements made in certain next generation sequencing techniques, and that leads us on to our possibilities section. Now, I've already told you how difficult it is to assemble whole genome sequences of T. cruzi, so a much better technique that we can use to begin to probe at genetic diversity is to do amplicon sequencing or Illumina deep sequencing. The premise of this technique is very straightforward. Essentially, what we do is we choose a highly resolutive genetic marker, i.e. one that has sufficient mutations or polymorphisms that can distinguish major parasitic lineages and potentially within a lineage. We perform an initial PCR using primers that, of course, are specific to our marker of interest, and then these primers contain unique barcodes at the 5' region. So these are unique strings of random nucleotides, usually between 6 to 10, and because we use these in our primers, they are incorporated into the amplicons produced during our PCR reaction. Now, we do this multiple times. We do this with essentially as many genetic markers as we like, assuming that our amplicons are of a similar size, either 250 base pairs or 500 base pairs. And then what we do is we pull our amplicons for sequencing, and we do this on an Illumina next generation sequencing platform. You can also do this technique on portable mini ion sequencing as well. Now, because our amplicons have these unique barcodes at the 5' and the 3' ends, we can essentially multiplex our sequencing to simultaneously detect multiple genes and multiple samples in a single sequencing pool. Depending on your sequencing depth, how much you're willing to pay for, we commonly sequence about 4 to 6 samples across 11 different genes of interest. And for us, commercially, it costs us about 50 cents per amplicon. So this technique is highly cost effective. And because we're sequencing a gene, not just assaying for the presence or absence of T. cruzi, this also opens up the opportunity to discover new polymorphisms in these genes, which may also be informative. And using amplicon sequencing, several research groups have begun to investigate parasite diversity in relation to clinical outcomes and to characterize diversity among different transmission cycles. I just wanted to highlight a couple of studies here that have used different genetic markers to probe at diversity within individual patients. And most report multiclonal complex parasite populations comprising of multiple different genetically distinct haplotypes. These studies are really beginning to reveal just how genetically complex individual Chagas disease infections truly are. The study here on the left, this used GP63, a surface protease, to examine parasite diversity in clinical samples in cardiac and congenital patients from Brazil and Bolivia. Here we can see diverse infections and in the principal component analysis, but what we don't see is any true association between parasite genotype and clinical presentation, for example. Similarly, here on the right-hand side, this used a different single copy gene that's genetically diverse across all parasite lineages. And what this study showed was the presence of multiple distinct parasite haplotypes in HIV co-infected patients in Bolivia. Now, these studies are a really good starting point, but they are based on just one or two genetic markers. And based on the genetic exchange mechanisms that I've already showed you, we can't rely on a single genetic marker to be truly informative of parasite diversity or to truly reconstruct phylogenetic relationships. Where this field is now expanding to, and really what I believe is now going to become, hopefully, the gold standard for T. cruzi genotyping, is to apply that same amplicon sequencing strategy, but across multiple genetic targets dispersed across the parasite's genome. With this strategy, what we're essentially doing is we're upgrading multi-locus sequence typing to become genome-wide locus sequence typing. And this study has done exactly this. It's developed this technique. This assay uses more than 200 genetic markers that contain regions of genetic variation. You can see them here. They're spread across almost all of T. cruzi's chromosomes. Each of these individual diamonds are areas of point mutations. And this study used this novel technique to begin to explore genetic complexity of T. cruzi samples from Colombia, from Venezuela, and from Ecuador in particular. Now, as well as being highly resolutive, this type of technology is also cheap. It costs about $4 per sample. Now, this field is only just beginning, but we've never really been in as strong a position with as advanced genotyping techniques at our disposal for T. cruzi than we currently are. And I'm pretty excited by the direction that this field is going in now. And this leads me on to my last bit, which really is where are we going with all of this? Is T. cruzi genotyping an area that we should still be investing our time and our research endeavors into? Well, I've only mentioned a few of the papers that have been using amplicon sequencing, but the more next generation sequencing that's being done, the more we're really beginning to lose any of those already loose associations of T. cruzi lineages circulating in different geographies and different ecologies. Essentially, what we're seeing nowadays is virtually all DTUs everywhere, all together, all at once. And what that simply reflects is just how much more resolutive these advanced genotyping techniques are. Suffice to say, we've come a long way since that Lancet paper in 1981. And what these maps on the slide show you here are the most up-to-date compilation of published T. cruzi DTU data. Basically, we see all of our lineages practically everywhere. And in some of the most recent amplicon sequencing studies, we're recovering parasite genotypes that are belonging to all DTUs, with the exception of TC-BAT, from single infections in our patients and our tradamine bugs and in our mammalian reservoir hosts. And really, these multi-clonal populations, they've been there all along. We just haven't really had the ability to detect them truly up until now. So having spent the past close to 15 minutes explaining T. cruzi nomenclature in quite great detail, what I think we actually need to do is almost scrap it all together and really redefine the way that we classify T. cruzi isolates and how we can approach some of these unresolved research questions which involve parasite genetics, given all of those inherent biases and limitations, particularly associated with obtaining good biological specimens. So I pose a simple question, which really is, to what extent are we limiting our thinking using contemporary T. cruzi taxonomy the way it stands? This concept of having 7 discrete typing units has certainly been a useful working framework for the last 10 to 15 years, but some of the assumed relationships and loose ecological and geographical associations that we have assigned to these DTUs, they simply don't hold true anymore. Now that we're able to characterize parasite genetic diversity at an unprecedented level, and our levels of resolution are ever increasing with our advancements in sequencing technologies and our improvements in genetic marker design. I personally think it's time for us to move beyond thinking of T. cruzi as these discrete individual typing units, which never really truly addressed the confounders of parasite multiclonality in the first place, and instead approach parasite genotyping using more of a metapopulation-based approach. So metapopulations, by definition, are spatially structured populations that consist of distinct units, subpopulations, separated by space or barriers, and that are conducted by our dispersal movements, in this case, our exposure events. And really, that's what multiclonal T. cruzi populations are behaving like, and they always have been. Anyway, that's my 2 cents, and I would be really interested to hear what everybody listening to the seminar thinks about this particular subject. And with that, I'm going to finish. So like I said at the beginning, I hope that you are all as enthusiastic about parasite genetic diversity, or if not, that maybe you now have at least a newfound appreciation for what is a really interesting and at times frustrating research area. So to summarize, T. cruzi infections are comprised of these highly complex, dynamic, multiclonal populations. Intra-host T. cruzi genetic diversity, it may play a role in diagnostic test sensitivity, drug response, and potential clinical prognosis, but our evidence is still very much indirect without strong causal inference. I've told you all about the various aspects of T. cruzi biology that are fundamental barriers to genotyping, especially the multiclonal nature of our parasite populations, our tissue sequestration in humans and animals, and low circulating volatile peripheral parasitemia. To date, genome-wide amplicon sequencing arguably represents the most viable advanced genotyping technique that's currently available to characterize parasite genetic diversity. And I would encourage anyone who is particularly interested in this area to read that paper that I showed you and consider implementing this methodology in your lab if it's feasible. And then finally, I leave you with that question of just how accurately does current T. cruzi nomenclature really reflect our understanding of parasite genetic diversity, and how can this be improved? And with that, I will be happy to answer any questions. Here's my contact information if you want to send me an email, and the link to my lab if you're interested in some of the ongoing research. Thanks.
Thank you so much, Dr. Messenger. That was a fascinating talk. Before I forget, I wanted to mention, because I've gotten a couple questions about it, that we will make your slides and a couple of these papers available to the USCN listserv, for those of you who are wondering about that. And I think we have time for a couple of questions. I want to encourage you guys to put questions in the chat, or feel free to unmute yourself and ask Dr. Messenger anything you might be wondering about. All
Thanks,
right.
Natalie.
Well, we have one from Jeff Whitman, who I know you know well, Dr. Messenger. His question is the following. He's very interested in T. cruzi sequencing work that can be done directly from host specimens. Can this quote unquote high def enough to sequence known antigens?
In theory, yes. Essentially, if you're able to design the appropriate primers and adapt the amplicon sequencing panel to the type of that part of the genome that you're interested in, if you want to sequence antigens, then yes. I don't know of anybody currently doing that, but I don't see why it isn't feasible from a conceptual point of view.
Okay. All right. If there are no other questions at the moment, since we just have a couple of minutes, I wanted to make a couple of announcements. One is that the next USCN virtual session will be on August the 4th, 2023. And then also that we'll be sending out a short electronic evaluation for this session. So if you guys could take a minute to do that, it'll help us be able to continue to offer CME for these sessions. So again, I want to thank you so much, Dr. Messenger. And we will be posting this recording on a couple of different sites. And so I'll be sending that information to the network hopefully later this afternoon. All right, so see you guys in August.
Thanks, everyone.